So in this video, I want to go through the cell cycle and DNA replication. And this will also sort of have to do with mitosis and meiosis, but the Mr. Anderson video in the mitosis-meiosis lab, I think that's lab 3, that I have linked in a different place in this document, is a phenomenal resource, so I don't really need to go through that, I don't think, because it's already done in a very succinct way. Here I have the cell cycle, which is the life cycle of the cell. And it has a few phases, but mainly you can split it up in a few ways. You have mitosis, which is the dividing of the cell, right? And you have two cells that divide to make a new cell. Then you have interphase, that growth phase, that place where the cell is growing and then ending up preparing for replicating. And again, focusing on our big ideas, right? Mainly in this, I want to look at regulation. How this is, at least for this part of the cell cycle, how this is regulated. And I have 3 points. These are often called the checkpoints. These are the checkpoints. Right, for the cell cycle. Right, and you have the checkpoint at M, the G1, and G2. So let's just go through basically what they are. We have this growth stage, and then it gets to this checkpoint. Right, so the cell has just divided, it's now growing, it's not replicating DNA, but it's going through transcription, translation, it's making proteins. Then the cell stops and asks itself, number 1, am I ready to start trying to divide? And number 2, do I want to divide? So this is, in a way, this point is sort of the point of no return. Right? This is the point of no return at that checkpoint. Right? So if the cell wants to start dividing, it goes past it, it's not going to stop dividing. Right? So all of these checkpoints, but this one, this G1 checkpoint, is really where the cell is going to decide, does it want to divide? Right? And how it will decide whether it's ready to start dividing, whether it's ready to start replicating DNA, and this is generally for for eukaryotes, because prokaryotes are just single cell organisms. But for eukaryotes, they'll get a signal, right? So at G1, they will get a signal, right, which is usually called a growth factor. Right? And then it goes through a signal transduction pathway. Right? So G1, right, the cell will get a signal, right, or this is our G1 checkpoint. Right? This is what's going on at the G1 checkpoint. For it to move on, the cell has to get a signal, right, from other parts of the cell, or sorry, from other parts of the organism. They'll get a signal through the endocrine system, right, and that there will be a pathway. And essentially what will happen, if we go back up here, we have a signal transduction pathway, we have a signal molecule that goes here. It will end up having a phosphorylation cascade. This active protein will be a DNA replicating protein, right? And so so signal transduction, and then your response is DNA replication, which I will get to in 1 second. But so this is like the big point. If it doesn't get a signal, if the organism is not ready for the cell to grow, or for the cell to replicate, then it will go through G0, which is sort of the static state, right? It's not growing, it's done growing, and it's not ready to replicate yet, so it's just going to stay here until it gets that growth factor, and that growth factor will tell it to start replicating. Once it starts replicating, you can't stop it. So DNA is replicated. G2 is the final growth phase, and it's preparing to divide. This G2 checkpoint is just, all this is it's asking, did the DNA replicate correctly? And it has some function, some internal function, where it checks if the DNA replicated correctly. It's not dependent on signals from outside of the cell. It just literally asks itself, did the DNA replicate correctly? And if it did, it gets past the checkpoint, right? This is the checkpoint that is subject to regulation from outside of the cell. The mitosis, it's the metaphase checkpoint, is between metaphase and anaphase. It just checks, and hopefully you know the process of mitosis. What this is are the chromosomes lined up correctly, right? So these are just important things that, right, this is how it makes sure everything's going right. These are sort of proofreading in a way. They're just making sure everything's going right. This is going to be the way that the rest of the organism is going to make sure that the cell is ready to divide before it starts dividing. Okay. The other part of this is DNA replication. Again, you should know how to do the A, C, T's, and G's. I doubt that will be on the exam, but who knows. But those are pretty simple. You can watch a video on that if not. I'm not going over that right now. The big idea of this, again, no details, the big idea is there are a lot of proteins, right? So if you want to disrupt it, you're probably going to want to denature. Denature proteins. As you can see, there's a lot of proteins here. If you're thinking about proteins to denature, what might have the biggest effect on this? I'd say probably look at helicase, or topoisomerase, or helicase. Or not helicase, they're polymerase. Polymerase, helicase, and topoisomerase. Those are going to be the ones that are like very, very big in having any of this happen at all. The Okazaki fragments are just a way that the cells can divide more efficiently by having lots of division going on at once, not just in one direction and then having to wait for it to go in the other direction, right? It's a lagging strand. Hopefully you're familiar with that. Really, the big idea, the one thing I could see the AP possibly asking about is some of these proteins that deal with the Okazaki fragments, disrupting, right, or denaturing one of them, and how does that affect the whole DNA replication process? And the answer is it will make it pretty much only work halfway, right? The leading strand will get replicated, but that lagging strand might not, or it could take a lot more time for that lagging strand to get replicated. So there's different steps of the process. Again, the AP may want you to know, if you disrupt one part, how will that affect the rest of it, right? But helicase, topoisomerase, and polymerase, those ones, right, are the big proteins. These two help unwind the DNA. This untangles it. This unzips the DNA. This makes the base pairs happen, right? So those are going to be the important enzymes in making sure this process happens smoothly and correctly. Okay, I'll see you in the next video.